Hello everyone. In this presentation we are going to study about disaccharides. Among oligosaccharides, 2 to 10 monosaccharides, then we call oligosaccharide. Suppose in this oligosaccharide, most important is disaccharide. If 2 monosaccharides, then we call disaccharide. Among disaccharide, 2 monosaccharides joined by a bond. This bond is called glycosidic bond. In disaccharides, again there are two subclassifications: reducing disaccharide and non-reducing disaccharide. Example for reducing disaccharide is maltose, lactose. Example for non-reducing disaccharide is sucrose. So we know that two monosaccharides join together by a bond called glycosidic bond. Okay, so let me explain how this glycosidic bond is formed. We will take two sugar. Maltose is a disaccharide made up of glucose and glucose, 2 glucose. Lactose is a disaccharide made up of galactose and glucose. Okay, sucrose is a disaccharide made up of glucose and fructose. So we will see the formation of glycosidic bond. For that I need to write the structure of glucose. I will explain with the structure of maltose that contains 2 molecules of glucose. Let me draw the structure of maltose by writing the structure of 2 molecules of glucose. As you know, glucose is a pyranose 6-membered ring. Let me write the number: carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6. Okay, so we know that in the 6th carbon atom there will be CH2OH. So we also know that if OH group is below the plane, it is alpha, above the plane it is beta. In maltose we have alpha glucose. Okay, we remember for beta glucose up down up down up, but here it is alpha glucose, so OH group will be below. So this is one glucose. So maltose is made up of 2 glucose. So let me write the structure of another glucose. This is also alpha glucose. When you write the structure of maltose, contains 2 molecules of glucose. When 2 glucose join together, we call glycosidic bond. Okay, glycosidic bond in case of maltose is alpha 1,4. Let me explain this is why. What is this alpha 1,4? This is the first carbon atom of first glucose molecule, and this is the 4th carbon atom of the second glucose molecule. So there will be a glycosidic bond between first carbon atom and the 4th carbon atom. When 2 molecules join together, so we will take out this hydrogen and we will take out this OH. So OH and so H2O, it will be released as water. Okay, so remaining is only O. Our glycosidic bond will be like this. Okay, our glycosidic bond will be like this. This is alpha 1,4 glycosidic bond. This is the glycosidic bond. When glucose and glucose join together, in case of maltose, it will form alpha 1,4 glycosidic linkage. Now let me explain here before going to reducing and non-reducing sugar. So this is maltose. You know, maltose contains 2 molecules of glucose, and the functional group of glucose is aldehyde group. So functional group is present in the first carbon atom, right? So in the cyclical structure, this will be the functional group or anomeric carbon atom. This is the anomeric carbon atom. Anomerism is always refers to the cyclical structure. This will be the anomeric carbon atom. Let me explain here reducing sugar. When in a disaccharide, if any one functional group is free, so in this case maltose, glucose and glucose, this functional group is utilized for formation of glycosidic bond. Any one of the functional group is free in given disaccharide, then it can reduce cupric to cuprous. So this is the Benedict's test. When you are going to learn the Benedict test, you will understand. Or even it can reduce ferric to ferrous. Then we call reducing sugar. Reducing sugar means in given disaccharide, if any one functional group is free, then they are called reducing sugar. Okay, as I explained. Let me take example of lactose. Lactose is a sugar present in the milk. It is made up of galactose and glucose. Here glucose can be alpha or beta, but galactose is beta galactose. Okay, we learned epimerism where we how to differentiate between glucose and galactose. Okay, let me write the structure of galactose here for you. We know that epimerism, glucose and galactose are epimers with respect to carbon number 4. If you know the structure of glucose, you can easily remember the structure of galactose. I said galactose beta, up, down, up. And let me number the carbon and it will be easy for you. Suppose this is a glucose, this will be up, down, up, down, up. We know that glucose and galactose are epimers with respect to carbon number 4. Here will be OH. If you know the structure of glucose, you can write the structure of any molecule. This is beta galactose because OH is above the plane. Then another glucose. Glucose can be alpha or beta. Okay, we will write beta glucose only. This is beta. Why? Because OH is above the plane. Here also the linkage is beta 1. Means there will be glycosidic linkage between this carbon atom and this carbon atom. Right? To make this glycosidic linkage. Here you take this OH and take this H and release as one molecule of water. Our glycosidic linkage will be, let me erase this one. Okay, so this is our beta 1,4 glycosidic bond. So here also if you take galactose is a aldose and glucose is also a aldohexose. So functional group is always present in the first carbon atom. Anomeric carbon will be first carbon atom. So this carbon atom is involved in the formation of glycosidic bond, whereas this carbon atom is free. So lactose is also reducing sugar. Maltose is also a reducing sugar. So you can see in maltose this carbon atom is free. Only this carbon atom, first carbon atom of the one glucose is utilized in the formation of glycosidic bond, whereas this carbon atom is free. Free aldehyde or ketone group in a disaccharide, then it will become reducing sugar. Now I will explain non-reducing sugar. Non-reducing sugar example you already know, sucrose. Sucrose is nothing but table sugar present in the fruits, okay, table sugar or sugar cane. It is made up of glucose and fructose, okay. So let me write the structure of glucose. 1, 2, 3, 4, 5. Glucose is here alpha glucose. Alpha means below the plane. OH. This is our alpha glucose. Fructose, we always represent fructose Purinose form. Okay, that means 5-member ring. It is called Purinose. So when you want to write the ring structure, ring form of fructose we already learned. It is the first carbon will be CH2 OH and the last carbon atom also CH2 OH. First will number. Okay, this will be the carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5 and carbon number 6. And the OH group here up. Whenever it is up, it is beta. Okay, up will be beta of fructose. I hope you understood the difference between alpha. If OH group is below the plane, it is alpha. If OH group is above the plane, it is beta in the anomeric carbon atom. Keto group is present in carbon number 2. And the glycosidic linkage in case of sucrose is alpha 1, 2. Actually, it is alpha glucose and beta fructose. So that means glycosidic bond is between this and between this. So you take this OH and this OH, you release it as water and you can join. So let me erase this one. You can join this. So this will be 1. Okay, 1 and 2. Here you know the functional group of glucose is always present in carbon number 1, right? Always present in carbon number 1. And fructose is a ketose. It is present in carbon number 2. Glucose is functional group is aldehyde. It is present in carbon number 1. Fructose first and second, the last carbon atom is CH2OH. The functional group is in the carbon number 2. Okay. So this is the functional group and this is the functional group. So the glycosidic linkage between first carbon atom and the second carbon atom. That means the glycosidic linkage in sucrose involves both the functional group. There is no free functional group. That means it cannot reduce cupric to cuprous or ferric to ferrous. So this will sucrose will become non-reducing sugar. I hope you understand the difference between reducing and non-reducing sugar. All monosaccharides are reducing because it has pre-functional group. If you take fructose, glucose, mannose, galactose, they are reducing because their functional group is always pre. In case of disaccharide, if you take example of lactose, so here one functional group is utilized here, formation of glycosidic link. This is galactose, this is glucose, the functional group is pre. If you take maltose, so one functional group is involved in formation of glycosidic liquid, other functional group is free. Whereas in sucrose, both the functional glucose is functional group is carbon number 1 and fructose functional group is in carbon number 2. So both the functional groups are involved in formation of glycosidic bond. It cannot reduce cupric to cuprous or cannot reduce cupric or ferric. Okay, this is a non-reducing sugar. Summary: maltose and lactose are reducing disaccharide, whereas sucrose is a non-reducing disaccharide. Okay. Thanks for watching.